Biology Colloquium. Our speaker today, Professor Jonathan Wendel of Iowa State University, is one of the world's foremost experts on plant evolution and speciation. His research focuses on how plant genomes and phenotypes diversify, with a special focus on whole genome duplication, aka polyploidization. Since receiving his PhD from the University of North Carolina in 1983, Jonathan spent most of his career investigating the genetics, phylogenetics, and phylogenomics of cotton, including four of the economically and culturally most important crop species on the planet, as well as 50 species of wild relatives in the genus Gossypium of the mallow family. Wild cottons exhibit tremendous diversity in floral form and habit, ranging from fire-adapted perennials in Australia to trees in Mexico. They're native to many areas around the world, including tropical America, the Caribbean, the Galapagos and Hawaiian Islands, Australia, South Asia, Arabia, and Africa. So reconstructing the evolution and historical spread of Gossypium has been a major challenge, and not least because of the widespread occurrence of hybridization and whole genome duplications in that genus. In pursuing this goal, Jonathan's lab became an international leader in using sequence variation in single and low-copy nuclear genes to reconstruct reticulate patterns of evolution in plants. And over the last several years, he's used these techniques to pioneer phylogenomic approaches to understanding cotton evolution. For his achievements in this area, including more than 340 publications on cotton alone, and contributions on many other subjects, including intergenomic, gene conversion, homolog expression bias, and the origin of plant form and diversity, Jonathan has received numerous awards. He's been named a Fellow of the AAAS, a Distinguished Fellow of the Botanical Society of America, Fellow of the National Academy of Sciences, and Fellow of the American Academy of Arts and Sciences. For many years, he served as chair of the Department of Ecology, Evolution, and Organismal Biology at Iowa State. Now, three little-known facts about Jonathan. First, he farms more acres of cotton than anyone else in Iowa. Actually, I don't know if anyone else grows cotton in Iowa, but with continued global warming, Jonathan, I'm not sure you can count on being the Iowa King Cotton for much longer. Second, Jonathan is almost surely fitter than almost anybody else in this room. Since 1997, he has run 158 marathons and ultramarathons, spanning all 50 states, totaling over 4,100 miles, roughly the distance from Ames to LA, back to Ames, and out east to Boston, where Jonathan fittingly had just been approved to run the Boston Marathon at age 70. That's wow, with several capital letters. But even Jonathan has to take second place to his wife, Kathleen, who has run even more marathons than he has, and including a super ultra marathon of 100 miles. Third, in my 39 years of being a UW faculty member, Jonathan is the only outside speaker I've ever hosted twice as a botany or biology colloquium speaker. And he's one of only 2 or 3 outside speakers overall in those series to repeat. That record is likely to stand. Now, it's not quite like being Steve Martin hosting Saturday Night Live 16 times, but hey, Jonathan, you're starting to get in that same neighborhood. So with all that in mind, please join me in welcoming Jonathan Wendel as our genial, generous, and genteel speaker, who will talk about genes, genomes, and the wondrous cycles of polyploidy in plants. Jonathan.
That was so wonderful and so embarrassing, and I'm afraid you're all...
Hold on a second, so we need to mic you. There we go.
Put this on where? But is this good? Can you hear me in the back? And this thing I put somewhere. Doesn't matter where. Well, Tom, thank you for that spectacular, generous, and yes, slightly embarrassing introduction. Genial? I wanted to work on... It's too bad this isn't a talk on gentians. I wanted to get that in there. Well, it's great to be here, and thank you all for coming. It's great to be back here. I gave my first talk here right before Tom joined the faculty, so it would have been about 40 years ago.
Wow.
I know, I'm older than dirt. I heard a wow. And then the mean age of dirt globally, I hear, is 42 years. And then I gave a talk here in 1998. I think maybe David invited me. And then I gave a talk here in 2012. The periodicity is this like an elliptic, about every 12 or 13 years. So you can all come back when I'm 83 for my next talk here. When I did give my first talk here, it was we had a genetic map in cotton. I had started to get interested in cotton. I was working along with John Dobley on maize in North Carolina, but I became interested in cotton because I was fascinated by the mystery of polyploidy. And I thought cotton would be accessible. We had a genetic map. We had 67 markers, morphological markers spread across 26 chromosomes. I could do something with that. And then fast forward 4 decades. And in my lab alone, we've sequenced 1,000 genomes, each a little smaller than the human genome. Wow, what a ride. And are we ever buried in data and opportunity and questions? And it's like every genomic rock we pick up, there's a new beast to describe. So that's what we've been doing for about 40 years. And while we've been doing that, the science is a community effort, so everybody else working on polyploidy has been doing the same thing in brassica or tragopogon or triticum or name your favorite polyploid, right? So today, I would like to talk about polyploidy a little bit. I see this as one of the singular realizations of the genomics era, that all plants aren't just polyploid, they're multiply polyploid. So I'm going to give a very general kind of talk. I hope it doesn't leave you too disappointed thinking it's rather shallow or thin, because I'm going to dance across sort of the high points of polyploidy and not show you lots of gels or slides or data. I'll be just talking about how people think in the community think about polyploidy and what we've learned so far, and give you a sense of where I think we're going. And I'll do that by using the genus Gossypium as an example. What do I have to do here? Where was I? Let's rewind in my brain. I think I was telling you what I was going to tell you. Yeah, I'm going to tell you. So I want to talk about what I view as one of the singular realizations of the genomics era in which we're fortunate enough to find ourselves, and that is that all plants have this history of whole genome doubling or polyploidy. And so although I'm going to give a lot of examples from my lab and from cotton, I'm really talking about all plants. So you can just think about your favorite plant. It doesn't even matter if you know it's a polyploid, because it is a polyploid. And so now you'll know that it is a polyploid. And so this is really a couple of well.
Is it not in PowerPoint?
Did that work?
Yeah.
What did I hit? I just hit one arrow.
Or hit the return button.
Let's see. No, the arrows don't work. The arrows on the keyboard don't work.
Just hit a return then.
All right, so the talk will consist of a couple of components. First, I'll introduce you to the genus Gossypium, shown here with this modern annualized version of the row crop. Return doesn't work either. That one works. Only the keypad works. Only the keypad works, but I can't go backwards.
Okay, this is a test.
This is a test. Okay. Tom alluded to this, and I was kind of amazed how much you knew about the genus, but it has a rather extraordinary evolutionary history. So I'll regale you with a little botany. And of course, I'm in a botany department, and as you know, nothing makes sense except in the light of evolution, and nothing in evolution makes sense except in the light of phylogenetics. And so I set off first to understand the evolution and the phylogenetics of the genus, so I have to introduce you to all of that. And then that sets up the story about polyploidy, because there's polyploidy in the genus, and I'll spend a good half my time today talking about the evolutionary genomics of polyploidy and what we, again, the community, has learned about polyploidy. So Tom already showed this slide. which is where the modern annualized form of Gossypium hirsutum grows. It's not native here. It came from more southerly climates, and after Europeans enslaved Africans and made it economically feasible to grow cotton and to further annualize it, it became the dominant plant in the southern states and in the economy more recently in one place in central Iowa. And from its New World location, cotton has spread worldwide to become the most important fiber plant in the world today. What I love about this slide, which I've surely shown here at least twice because I took it 30 years ago out of a Land's End catalog. You can't see here, it says cotton, one of Egypt's greatest treasures, but this is a New World plant that was domesticated about the same time those buildings in the background were being built. So that's the history of crop domestication as an example right there. What's remarkable about cotton is that this kind of evolutionary transformation under domestication from the photoperiod sensitive tropical antecedents on the left that have very few flowers, they're not synchronously blooming, they have crappy little short brown fibers that are tightly adherent to the seeds, became transformed by about 5,000 years of strong directional selection by humans into these annualized row crop plants you see there on the right with their strong, long, fine white fibers yielding abundant lint, synchronous flowering, et cetera, et cetera. Incredibly, this happened 4 times, this transformation shown from wild forms to cultigens, to land races, to modern varieties like the 18-inch tall ones from Oklahoma there or West Texas, shown on the right-hand side, the storm-proof sort of annual forms. This happened 4 times from 4 different wild progenitors, twice in the Old World, twice in the New World, all involving approximately the same morphological transformations. And so naively, I said a long time ago, ooh, wouldn't there's how evolution makes a new structure. I've got this great system. I could go figure it out. And I have a 5,000-year-old experiment. Graduate students can't hang around that long. We can do it in the lab. We have the antecedents and we have the descendants, and we can make crosses, and we can get RNA and DNA and proteins and metabolites and everything out of the fibers, and we can try to learn about the evolution of morphology. We're barely scratching the surface of that. That's for all of you to do. It turns out that evolution is still rather complicated to study. This is what a wild cotton plant looks like. About 6 months after a hurricane, this is quite topical today, about 500 meters from the southern tip of the Everglades in Florida. And it doesn't look much like a row crop, does it? There's these incredible perennial trees, small trees and shrubs. And look at the capsules there. And the seeds, that's my hand for scale. And those tightly adherent, single-celled epidermal trichomes that humans, again, 4 times, 3 of them shown here, transformed into the row crop forms up on top. Amazing sort of parallel evolution. And we're attacking this right now to ask about this at a really fine scale, a genomic scale. How convergent are they? At what level do we mean convergence, et cetera? It turns out these are all rather quite different genetically in what's going on, how they're tweaking that massive co-expression network that directs development of the primary and secondary cell wall. But anyway, so there's this parallel domestication of this trait for which cotton is very famous. You can see, I'm in a botany department, I can say ovule. And you'll notice that without explaining, that on the surface of the ovule you find cells, 1 out of every 3 or 4, that's terminally differentiated into a slated to become a trichome on the day the flower opens. I don't have to explain anthesis, woohoo! And then 3 days later, it started to lay down the primary cell wall, which happens for 2 weeks in the wild forms, about 3 weeks in the domesticated forms. There's been a prolongation of elongation. And then it becomes a cellulose factory. And it starts to fill the lumen with nearly pure cellulose, over 98% cellulose. There's a whole molecular machinery involved in this, and it's really interesting and complicated. And it takes about 2 months from anthesis to maturity, and then it forms the lumen collapses, the cell dies, and it forms these flattened spiraling like ribbons that will therefore stick together and be spun into yarns and threads, and that's what gives cotton its special properties and makes it such a wonderful plant for commerce. Okay, this is where we find, oops, I can't go backwards, sorry. Oh, I wonder if I did. No, I don't dare. This is where we find, Tom mentioned where some of the diversity in the genus, this is where they grow. I've been to many of these places, not all of these places. There's still a tremendous amount of diversity in the southern part of the Arabian Peninsula and in the Horn of Africa, and I can't really go there collecting. So I like to say there's 50 species in the genus. I'm sure there are a lot more, but this is where the exploration really needs to be. I've been to most of the rest of these places to collect these. There's really extraordinary diversity in the genus. Most of them are shrubs that grow in dry places. Gossypium australis, shown in this slide, in the middle of the Australian continent. You now know we're not in the middle of the Australian continent. You can tell there's a characteristic floristic marker right there, but you can, of course, you were bored by that and you started fixating on the Gossypium harknessii here in the foreground. And then, of course, you can find the Gossypium in this picture if you look really closely. Oh, wait, no, I'm kidding you. That tree, that 10-meter tall tree on the western slopes of on the western side of Michoacan and Oaxaca and Guerrero, there's a clade of arborescent cottons, and there you can see it's in full flower. I don't have a pointer. I'd point, you can see these flowers this big at the tips. It flowers when it's naked. So there's just amazing diversity.
There's a stick, by the way.
Yeah.
Pointer next to you.
There's a what?
There's a pointer on the wall.
There's a pointer?
A stick.
A stick. Ah. I don't know. It's pretty old school. I haven't used one in a long time. But so this is what the kind of diversity we see in the genus. And doesn't that make your heart do a little pitter-patter? It makes my heart do a little pitter-patter, apparently. And so we wanted to understand this, and so we started working on the phylogeny of Gossypium. And this reads like a history of molecular systematics from, oh, I know, let's quiz the audience. Who remembers allozymes? Raise your hand. Okay, RFLPs. John Dobley does. AFLPs. Ooh. ISSRs. Oh, the I-omega zip drive of molecular technology. Single copy nuclear genes, chloroplast DNA restriction site analysis. And now, of course, whole genome sequencing. And we now have a really good understanding, I think, of what the evolution of the genus looks like, and it looks like that. And we have what we call genome groups. These are all monophyletic. These are monophyletic clades that differ in genome size and chromosome behavior and interspecific hybrids and that sort of thing. We know when the genus originated. It's about 5 or 10 million years ago, and so now we can use a phylogeny to do all sorts of interesting things. We can look at genome evolution, and all these are diploids, and they have 13 chromosomes. But they vary a great deal in chromosome size. From the tiny little genomes, this is the first one we sequenced, of course, back when sequencing was really hard, to almost a threefold difference in the ant-dispersed clade from the Kimberley region of northwest Australia. And it turns out that's a subject for a different talk, but this represents the dynamics of the transposable element fraction of the genome, the differential proliferation and differential deletion of different classes of transposable elements at different periods during the radiation of the genus. But apart from that, the chromosomes throughout the entire genus are largely collinear. There's just a few rearrangements with the same basic 30,000 genes or so, a little more, maybe 34,000 genes in each of these on the same chromosomes. But they differ threefold in genome size. We can diagnose multiple instances of transoceanic dispersal. Cotton has a real fondness for oceanic voyages. The genus is only 5 million years old or so, and look at its distribution. It's quite incredible. We don't know how this happened, but since I'm at the University of Wisconsin, can't I invoke Hugh Iltis? I think he was the one who blamed the constipated duck for all of these long distance dispersals. Am I getting that right, or was it someone else? Who remembers? Was that Hugh? I didn't know I was going to say that. That's nice to be able to be here and say. There's Ingrid just pointed out we have this lovely plant, this lovely polyploid that's out that made it out to the world's most isolated oceanic archipelago, Hawaii, where Gossypium tomentosum is just doing fine. Thank you very much. There's two different dispersals of the Galapagos Islands. A very important one to Peru, which gave rise to one of the two donors, the lineage that is the closest relative of one of the two donors to modern allopolyploid cotton. And then we had that one. And that's the one that's most important for today, because that's when the constipated duck carried the A genome propagule across either the Atlantic or the Pacific, we don't know, where it encountered an endemic D genome species. Even though they've been isolated for 5 million years or so, having evolved a twofold difference in genome size, they said, let's get together and let's make babies. And they did, and this gave rise to a new lineage of allopolyploids, and it subsequently radiated to the 7 species we now recognize down below. Later, humans evolved, and they found 2 of these species, Gossypium barbadense and Gossypium hirsutum, and decided to bring those into cultivation, and those gave rise to the 2 modern crops that dominate cotton commerce today. Hirsutum, which is upland cotton, the cotton of t-shirts and blue jeans and Q-tips and all that. And your money, by the way. That money sign in Iowa is because about 50% of your dollar bills are cotton. The mint won't actually tell you the percentage. That's top secret. Anyway, so these are the lineage of polyploids. Paralleling this development in our evolutionary understanding of the genus and its diversity, the genomics era happened. So this now feels antediluvian. 12 years ago, this work started 15, 16 years ago, we started to sequence the smallest of the genomes, the D genome progenitor of allopolyploid cotton, Gossypium raymondii from Peru, an almost extinct plant, by the way. And it was a huge effort. And since then, the floodgates have burst, and genomes started appearing. We've been involved in a bunch of these, but now there's a huge effort among colleagues, many of them in China, and the number of genomes just continues to blossom. And there's at least 125 de novo full assemblies, some of them really great telomere to telomere assemblies that we have now. Pangenomes for all these different groups. There's, what happened there? Wow, well, I think what it says, genomic and transcriptomic resequencing, we have at least 5,000 resequencings data sets for diploid cotton, over 15,000 for allopolyploid cotton, because it's such an important plant and so many people are doing interesting things in it. So that was the introduction of Gossypium, and that'll lead me to talk about polyploidy. But to summarize, this is all I've said so far. There was a common ancestor 5 or 10 million years ago. It diverged into an African and American clade in part. I'm just showing you the parts that matter for now. And each of those have about 35,000 genes that are largely collinear, but they exist in they have a twofold difference in genome size. And I like to think that evolution finely tunes these 35,000 genes to sing in a perfect 35,000 part harmony, and that my song is going to be different than someone else's song who's across an ocean for 5 or 10 million years. So to me, it was absolutely stunning that polyploids were even possible, that you could bring all these, that you could double the gene complement in only one of the 2 parental cytoplasms, change all the intracellular stoichiometries and volumetric relationships that are involved in metabolism, and have it work. How can that ever happen? But it's not just in cotton. This is super common in plants. So I just said, huh? And I had this framework for studying it, and we now decided to start very simply with what we could at the time, which was looking at single copy genes and repetitive DNAs, and we started comparing the duplicated products that result from polyploidy. These are called homeologs, to each other and to their parents using our phylogenetic understanding of the genus. And this was just what we could do at the time. We started looking at single copy genes, we started looking at gene expression, so on and so forth. And as to reiterate, this is not just my lab doing this, it's everybody who's working on polyploidy globally. We're all doing this sort of together, and we're all reading the same literature and seeing what we can learn. And so now I'm transitioning in this talk to what have we learned about polyploidy. And again, all I can do is touch upon some of the high points. And that, I'm sorry to say, we'll keep it at a somewhat trivial level because biology doesn't really operate at the single gene level very well. I mean, metabolism, extraordinarily complicated phenotypes. The genotype to phenotype equation is still, I mean, that's the future. We don't know much about it right now. That's where we're all trying to head. But I can tell you what we've learned so far, at least about a couple things. So I'll tell you what happens to genomes, so what happens at the DNA level, and I'll tell you a little bit about what we've learned about what happens to gene expression. And maybe at the end or near the end, we'll touch upon some of the other dimensions. Okay, so this is what we've learned at the DNA level. Gregor Mendel has an evil twin, and he looks like this, and that's his name. Non-Mendel. And so using this idea, using this simple phylogeny and this expectation that comes from an evolutionary understanding, we started finding all the ways in which this null hypothesis, this expectation of what one should see understanding the organismal history, has been violated by different forms of non-Mendelian, intracellular, intergenomic behavior, such as gene conversion between two sequences that are collinear but in different genomes. So if you take an A genome which has a blue gene and a D genome which has a pink gene, in the polyploid they could interact perhaps and give you chimeric sequences. Maybe they can rescue deleterious mutations, et cetera, et cetera. Nobody knew, so we started looking. It can be a diversity eliminating process. That's the one I mentioned, rescuing mutations, for example. And we find it. We find it, for example, it's not, I would say it's not super common in the community, but it does happen. It doesn't happen that much in Gossypium, I think probably because this twofold difference in chromosome sizes prevents more interaction among homologous chromosomes than might be in some other polyploids, although at the chromatin level, at using conformation capture technologies, we do see homolog touching. Anyway, if you think about the fact that you have a mom and a dad and both are donating nuclear genomes, but only one of them is donating organellar genomes, you might expect cytonuclear gene conversion. Did I tell you? All the New World cottons have Old World cytoplasm. They all have an A genome mitochondrial genome and an A genome chloroplast genome. And so for some systems, not all, in fact, I would say, let me correct myself, few systems, we find that you get maternally biased gene conversion, where the sequence, the nuclear gene encoding a product that ends up in the plastid becomes converted, and also bias favoring transcription of the native A genome copy. So that's one way, gene conversion. Another way this hypothesis can be violated is unequal rates of evolution. And here's some data for two different genomes where we see, I guess this is 700 genomes that we sequenced. So this is all the genes that, genic, not non-genic. So they're purely homologous, and in this case, orthologous copies. And you can see that the A homologs in Barbadense and the A homologs in Hirsutum have higher nucleotide diversity than in the D homologs, even though they're in the same organism, in the same environment, experiencing the same external selection pressure. But this is going to say something ultimately about how selection and homologs interact and uniquely in their each environment. So the selection is seeing the two genomes rather differently. Probably selective sweeps are different in the two genomes, for example. So you get unequal diversity. That's an asymmetry between the two genomes. Gene conversion can be asymmetric. Diversity accumulation can be asymmetric between the two genomes. And I want to make the point before I forget that asymmetry in the evolution of the two co-resident genomes in allopolyploids is a very common observation now. You see it a lot, dominant genomes and terrible words like submissive genomes, but you find asymmetry in genome evolution following allopolyploid. You even see it in the accumulation of deleterious mutations in cotton at the population level. A very complicated slide, but at the bottom means fewer mutations. At the top means more deleterious or slightly deleterious mutations. And the colors represent here 6 different species. So blue is higher than blue, purple is higher than purple, yellow is higher than yellow. Across the board, the A subgenome accumulates more deleterious mutations than the D subgenome, and in all branches of the phylogeny. So that's that. We expect gene loss, right? Because of the time of gene duplication, and the race is now on. You would see that the race is on to either find a new job to do, or partition the old job, or be obliterated by mutation, right? So you expect to see a lot of gene loss following polyploidy, and it's comforting that we do. Here's a phylogeny with an original, this is an older set of data, but all the new data are the same. You can see that there's a little more gene loss among the diploids in the D genome than in the A genome, 390 versus 234. But in the allopolyploids, gene loss not only is higher, but it's again asymmetric. It's biased towards the larger genome as losing more genes than the smaller genome. So gene loss is more frequent in the allopolyploids. It's asymmetric, and it's the same pattern for all species and branches in the polyploid clade and in more recent studies of the same phenomenon. And then we can get intergenomic transfer sequences. Obviously, transposable elements can spread following polyploid formation, and I'll say a little bit more about that now. The second form is a little more recent in how important, in the recognition of how important it's been in allopolyploids writ large, and that is homologous exchange between the duplicated copies of all the chromosomes. So I'll walk you through this. There's an allopolyploid with 2 genomes, grayish white and red, and it can look pretty much like cotton. We don't have many homologous exchanges. We have somewhere between none and a few, depending on the data set. They're pretty stable at the chromosomal level. Other polyploids have a lot of homologous exchanges. They get crossovers between the homologs, and they can start to scramble the sequences. In fact, in some of them, if you look a little old, if you look in some polyploids, some of them are really biased towards the survival of one genome preferentially over the other. Now you can play this out. Here's a possible outcome where the one subgenome is retained following homologous exchanges over time. And the poor systematist or evolutionary biologist or genomicist comes around, looks at that and says, oh, look, I found a new autopolyploid. It's not an autopolyploid, it's an allopolyploid. And so this is one more way in which those terms are wholly inadequate. We've known they've been inadequate in lots of ways. But this is a sliding scale in both directions. And we can talk, it would take me a whole seminar to talk about all the different ways in which you can transition between autopolyploidy and allopolyploidy. Why am I talking about this? Because it's everywhere now. It doesn't, you can't throw a rock at the literature. Don't throw a rock at the literature. You can't pick up a polyploid these days without finding something about homologous recombination. In fact, here's an example from, even if you take subspecies of rice, Indica and Japonica, cross them, I was lucky enough to be involved in this collaboration, cross them, make an allopolyploid, single seed descent for 4 generations, sequence them all, and make Indica green and Japonica red, their chromosomes. So they have 12 chromosomes, and sequence 36 different lines and look at all the greens and reds resulting from homologous recombination. It's recombination gone wild, and it also matters. So you can ponder that next time you go to your coffee shop and have a double. espresso and realize that the reason that your coffee tastes different than my coffee isn't because of genetic diversity, it's because of genomic diversity at the level of homologous exchanges. This is a new set of papers that was out this week. Okay, that's my whirlwind tour of what the community has learned about some of the fundamental processes that happen to the genomes of allopolyploids following their formation. I have to give an equally quick tour of what happens to gene expression. When I started this work, nobody knew how many genes were expressed in a single plant cell, let alone what would happen to duplicate gene expression. Can any of you now tell me how many genes are expressed in a single plant cell? Well, there aren't many single plant cells until recently. People weren't doing single cell sequencing. Right now it's common. Well, it's getting more and more common. But nobody knew. We thought maybe 5,000. An individual diploid cotton will have about 25,000 genes expressed in its fiber, which is a single-celled epidermal trichome. Okay, so about 50,000 genes are expressed in a single allopolyploid cotton fiber cell. About the same number from each of the two. It's a little different, but about the same number from the two homologous copies. How are they each deployed through development? Everything's doubled now. How are they used? So, around 1998 or 7 or something, I said to NSF, huh? And they said, go figure it out. And so we started doing what we could at the time, which was, this is the only gel I ever show anymore. This was Keith Adams came to my lab, and we started working on using single-strand conformational polymorphism analysis, where we could take the cDNAs from the D homolog and the A homolog for one of the 7 genes that encode alcohol dehydrogenase in diploid cottons. All 14 are retained in allopolyploid cotton. And you could go into the cold room with acrylamide and 8 molar urea and run these horrible gels, radioactive, and then measure, and then you could see the proportion of the transcript pool for this one duplicated gene that came from each of the 2 genomes. And that's what these numbers are. These are all proportions. And these are all the tissues that we looked at. And isn't it fun to look at? Look at all the variation from tissue to tissue to tissue, just for one pair of homologous genes. Expression varies that much just due to what's going on in the organ or the cell type at that time. Look at that, and just look at the floral whorls there. You have, maybe I said put that there. You have reciprocal silencing of alternative homologs during development. Gene expression is so very interesting, and so everybody's been studying this, and one of the commonalities that everybody finds is biased expression. At the gene level, it looks like this. Instead of just getting 50-50, green and blue, you can get mostly blue or mostly green. That's called homolog expression bias, and that's very common for genes, and it can vary from cell type to cell type. It varies from cell type to cell type. I would go so far as to say there's no gene in any plant that's expressed equivalently in all tissues. We can't find one, and I bet it's really hard to find. Oh, you could probably go find one, but it just means you haven't looked hard enough at more cell types. And you can also have, at the genomic scale, you can have preferential transcription of one genome over the other. And this can vary. So you see at the top one, it's balanced. It varies by gene, but it's 50-50 overall with respect to color. And the bottom one is unbalanced. We're mostly blue. One genome is being transcribed more than the other during development. We don't know what this means. It's true for cotton as well. Here's a bunch of tissues at the bottom. Wherever you have an asterisk, you have statistically different levels of gene expression between the 2 genomes. So this is now a very common observation. It's often framed, this kind of thing is often framed in terms of subfunctionalization or neofunctionalization of duplicated genes following whole genome doubling. Jim Birchler here has written perhaps most extensively about what happens following gene duplication in plants. But if you just go to the literature, this was yesterday or the day before, I just looked it up again. It grows by leaps and bounds. And you put in something like polyploidy neofunctionalization, you'll find over 10,000 citations. People are super interested in this, in neofunctionalization. That's what it looks like sort of schematically. This is where the two do, these are the ancestral copies. One of them gets a new job. That's neo-functionalization. Or they partition the aggregate older job into different organs or tissues, and that's subfunctionalization. And people are very interested in this because neo-functionalization and subfunctionalization are often show up in titles that have to do with stress and agriculture. Heat stress, drought stress, water stress. Transcription factors are often involved, and they tend to be retained at a very high level and evolve very quickly following polyploidy. So that's a lot of the intellectual framework for this kind of gene expression evolution. This is not cotton. I don't even remember what this is. I think this is zebrafish. But I just wanted to show a very simplistic diagram of why what I'm talking about is way overly simplistic. Genes don't operate in isolation. Genes operate as part of complicated gene co-expression networks with complicated underlying biophysical and biochemical and evolutionary dynamics. And so what we really want to do is start looking at co-expression networks of genes. And we're doing that, and the community is doing that. And it just doesn't lend itself well to an overview kind of talk. But here's just a couple of early efforts. So in cotton, if you'd like to know, the two genomes have similar yet different co-expression networks. It's really interesting when you put the co-expression networks together in the same nucleus, you start asking what percent of the homologs show up in the same module of co-expression versus not. What happens to them under directional selection? So here they respond differently to directional selection. Selection, in fact, really under domestication, a lot of the modules really form. The hairball, which is really diffuse, gets a lot tighter. It's really interesting. That takes you a little closer on this pathway from genotype to phenotype. And they can respond differently to ecological conditions, to salt stress, for example. Uh-oh. Oh, there we go. Okay, and so that's my very quick, simple introduction to what happens at the DNA level and the RNA level in polyploids. And if that's not complicated or mysterious enough, I'd like to tell you that I've pulled the wool over your eyes. And I really have not been talking about polyploidy. If I take you back to the very first paper that we wrote in 2012, the first two words were repeated polyploidization. And I know this is going to resonate with some of you in the audience because you know about the multiplicity of polyploidy events in history. And here's what it looks like. And this is now to wake you up. This is going to be the audience participation arithmetic part of the talk. So at the base of the flowering plants, there was a whole genome duplication. And at the base of the seed plants, there was a whole genome duplication. And so let's count genomes together. What's 2 times 2? 4. Ooh, I have a responsive class. And then at the base of the eudicots, there was a whole genome triplication. So what's 2 times 2 times 4? 12. Not always the same hands, please. And on that triplication here, you see little rainbow phylogeny giving rise to two of my favorite genera, the wine genus and the chocolate genus. So now we have 12 genomes. And then, we had the evolution of Gossypium in this part of the Malvaceae. And there was a 5 or 6-fold multiplication. Maybe it's 5, but I showed it here as 6 because I don't want to redo the slide, and it gives a bigger number. So what's 2 times 2 times 3 times 6? Biologists, not mathematicians? What's going on? I get 72, am I right? Yeah. 72 genomes in, diploid cotton. Yeah. Which means the one I've been telling you about has 72 times 2 is 144 genomes sort of ancestrally in a cotton plant. Now play this out throughout the whole phylogeny of angiosperms. In your head, how do we know this sort of thing? It's complicated, of course, but comparative synteny is is an important tool for this. Here's a piece of the grape chromosome aligned to a piece of the cacao genome, Theobroma chromosome, and here's some remnant sections in diploid cotton that align to that. And you can do this throughout the whole genome, and you can try to reconstruct how many genomes were in the ancestor. And there's a number of techniques, and it's an evolving field how to do this and how to have confidence in it. But it's reiterated throughout angiosperms. Oh, but I should have made the next one spin, because this paper just came the next animation should spin, because my head is spinning. The genome of okra just came out, and what did I highlight there? We found that okra has experienced 3 additional whole genome duplications compared with diploid cotton Gossypium remundii. So 72 times 2 times 2 times 2 is 432 genomes. That'll make a snotty mucopolysaccharide-filled vegetable, won't it? 432 genomes. Wow, isn't biology and evolution wonderful? And try to go figure out the dynamics of the survivorship of the genes here and what all that was about, and reconstruct the history. That's just fabulous. So polyploidy is ubiquitous, it's everywhere, it's recurring. It's not just in the classical allopolyploids, but it's throughout angiosperms, throughout the phylogeny and evolution of angiosperms. Old plants that we used to think were diploid, like the maize here. I think this is John Dobley's slide picture. No? Who did it? Hugh. This is how wonderful that I get to use Hugh Iltis's slide. Maybe I should attribute it, or I should just keep stealing it. He wouldn't mind. Hugh Iltis's slide. These are not diploids. These are all paleopolyploids. Diploid cotton is not a diploid. It has 72 genomes. The average angiosperm, this is a brand new paper, just came out in American Journal of Botany. Mike Barker and Mike McKibben just published this. This shows bar graphs showing the number of species and the number of genomes that are inferred, the number of doubling events that are inferred to have come in that lineage. So there's a mean of about 3.5 doublings in every angiosperm plant. There's so many implications of this, it's hard to know which one to start talking about first. Pick one. You can pick, for example, genome size. This is a bar graph of about 2,500 angiosperm genome sizes that shows most of them are small, a few are really big, some famous ones like Paris, or what's the new, the Mesipterus, just got published earlier this year, gigantic genome. There's the mean over there, about 2.5. That says picograms, not too different from gigabases. So take, there we go. Take the ancestral angiosperm genome size, which may be estimated at 300 or 400 megabases. Maybe it's a little bigger. It doesn't matter really how much bigger you make it. The point will still be the same. The actual number will be different. Multiply by 144 genomes and predict that upland cotton should have a pretty big genome. Let's just say 50 gigabases. But what is it? It's 2.4 gigabases right here. Well, in other words, it's lost a lot of DNA. So following each of these cycles of polyploidization, there's a lot of jettisoning of all this extra DNA. And it's very interesting to think about, how that might happen mechanistically. What does evolution see? How is anything retained in dealing with all the gene disbalance, gene unbalance that happens during this process? What about chromosome number? Same thing. If you take the ancestral chromosome number of angiosperms, 5, 6, 7, whatever, do a little algebra, you get 1,008 chromosomes predicted, and it doesn't have it. Cotton is 2n equals 20, 2n equals 4x equals 26. So, so there's a lot of chromosome number condensation. And this is what I've called the part of the wondrous cycles of polyploidy in plants. You start with whole genome doubling, and all these processes are set in motion that I talked about very briefly at the DNA level and the RNA level. But this is happening while there's massive genome downsizing. And it can be asymmetric. In this case, I think the B genome is being pockmarked a lot more than the A genome. And here's an example just from the most recent neo-allopolyploid event in cotton. the cotton I was talking about until just a couple minutes ago, where I introduced all the other genomes. This is where the A genome and the D genome hybridized, and they gave rise to the allopolyploid AD genome. But do the arithmetic there. Something's missing. About 200 megabases, right? And we're beginning to know quite a bit about it. And I mentioned earlier that I would talk a little bit about transposable element proliferation following polyploidy. It turns out there's some genome size equilibration going on subsequent to polyploidy. We haven't gone in and really worked out the absolute details of this yet, but it's an obvious study just waiting to happen. The A genome is getting smaller. It's lost about 200 megabases in spite of the D genome getting a little bit bigger. And this is in the face of bombardment of each other. The A genome, which is more gypsy-rich, has been spreading gypsy retrotransposons genome-wide, and the D genome, which is more copia element-rich, has been spreading copia elements throughout both genomes. But on balance, the A genome is shrinking and the D genome is growing. Okay, and that's biased fractionation I've already mentioned. And this is true in cotton too. Cotton has, although it hasn't had a lot of gene loss, it's following the generalized rules that are out there. So genes that are involved, genes that encode proteins that function in organellar processes or that are imported into the proteins imported into the organelle, they're restored to single copy, sort of preferentially. Or those involved in DNA repair are restored to single copy. Okay, that's what I just said. And then there's a lot of chromosome number condensation, reduction. We don't have 1,008 chromosomes anymore. And then for different lineages at different times throughout the whole angiosperm phylogeny, it happens again. And the whole wondrous cycle repeats itself over and over again at in different lineages at different times, idiosyncratically, and in some huge lineages, rarely or not at all. My good friend Jeff Doyle always says to me, "Why do you keep talking about polyploidy? It almost never happens in faboid legumes." Right? And that's pretty species. That's what happens, but given the species diversity of legumes and how old they are, not very much. Why is it common in some lineages and uncommon in others? We don't know. So why does Solidago go hog wild with polyploidy and other genera doesn't touch? Okay, well, I'm almost done, I promise. I know you have questions, and you might, and it depends on how you're oriented. So if you genomically, mechanistically oriented in your thinking, you might wonder, what is it about the immediate consequences of genomic merger that were set in motion by history of the organisms involved? Soybean does not have unbalanced, it has balanced fractionation. Maize has highly unbalanced fractionation. You might know about M1 and M2 as they're known, the two genomes in historically allopolyploid maize. And we can talk about whether transposable elements and their proximity to genes set certain dynamics in motion in terms of how selection might operate or not. If you're more pop-gen or ecologically oriented, you might wonder how mutation, drift, and selection interact in different and unique ecological settings to bias retention or loss of certain genes. And there's a little headway being made here. It's tough stuff because you have to know a lot, and you have to do a lot of genomic resequencing of populations of polyploids compared to diploids. And that's tough. There's a lot of groups, not a lot, but a number of groups in Europe that are doing some pretty good work on cardamine and Arabidopsis and genera like that to look at the efficiency of purifying selection and whatnot in polyploids compared to diploids. In cotton, we know that there's very different efficiencies of selection. Deleterious mutations accumulate more, as one might, perhaps not surprisingly, in polyploid cotton. How does this view of plant genomes change the way you think about plants? I mean, to me, it's wonderful just to know, but it's also, I think, kind of important. Like, I remember back when we first could do cloning and sequencing, we started amplifying MIB transcription factors. And we would amplify them and clone them and sequence them, and every one we sequenced was different. And we stopped at around 300 different MIB transcription factors. And I couldn't figure out, how did plants end up with so many MIB transcription? I mean, humans have like 4 or something like that, right? And they didn't have any special means of spreading around the genome. They don't need special means. They just need to be preferentially retained following 144 genome duplications, that sort of thing. And the fact that they're there also changes my view. That means perhaps they're doing something in terms of adaptation, either to climate or to stress or to morphology with their differential retention. So I think knowing this about plant genomes, you can actually kind of go backwards, look at the genome, find something that's doubled or more, and ask what is it doing as a means of discovery. And then how does polyploidy generate ecological novelty, morphological novelty? How is it that 1 + 1 does not equal 2? And here's what an A genome and a D genome fiber look like compared to an allopolyploid fiber. And these are all just single-celled trichomes. And I've already told you how complicated this is with 50,000 genes expressed in this one single cell. I got into this so naively, but oh my goodness, what a labor it's been, and what a labor of love I hope it is for all of you, because what's going to be required is going way beyond what we can do now to try to synthesize this whole G to P connection here, this whole genotype to phenotype connection. We've made temporally dense transcriptomic analysis of the developing cotton fiber, both for barbadense and hirsutum. At the same time, we've done the whole carbohydrate map, glycomics of the developing cell walls. At the same time, we've looked at proteomics of the developing cell fiber. You have to put together all of these omics along with experimental populations to try to make some understanding of how it is that you can get these different morphologies from basically the same fundamental toolkit that basically evolves this difference over 5 or 10 million years, and then this more radically and suddenly with polyploidization. So it's complicated even for a single cell, but I think it's a worthy venture, and I think that's where the community is headed. It involves all kinds of tools, not to figure out what's going on epigenetically, and I mentioned chromatin conformation maps and all of that, to put it all together, to try to understand how that whole network that leads to morphology is tweaked by something we also care about, which is selection and evolution in nature, and how humans practicing strong directional selection on mutations they don't see propagates up through this whole mesh of networks and omics to give rise to a transformed morphology. So I think I will leave you with that thought, except the most important part, which is to thank all of my mentors, colleagues, and nearly 40 years of students and postdocs and collaborators, and sheer privilege of being involved here and being born at the right time and place and having colleagues around the world to share this journey with. Oh my goodness, what a lucky man. But that's my story, and here's what their names are, some of them, especially my longtime collaborator. She was my undergraduate first and is now an associate professor in my lab, and my ex-student Guanjing, who runs a big lab in China now. She's a genius, and I recommend you all go work with her too. So thank you very much for having me, and I'll be happy to answer any questions if I left you with any. Did I leave you with any questions? Or are you shell-shocked?
So in recent years, a fellow by the name of Adam Roddy has proposed a connection between genome size evolution and photosynthetic rates. That basically, in order to achieve high photosynthetic rates, small cell sizes are required in order to make venation at fine scales and small mesophyll cells that would effectuate high rates of photosynthesis. So I'm just wondering, with cotton and with some of these rather large genomes, have people looked at the relationship among them in terms of photosynthetic rates? I mean, I can imagine with some of the more dry land species that they have rather low rates as a possibility.
I love the question, Tom. What we haven't done, you have arid zone plants in the diploids and in the polyploids, but we've never surveyed, say, epidermal cell sizes, guard cell sizes, and no one's ever done that experiment to actually look at the correlation between genome size and photosynthetic rates and cell sizes. There's a strong nucleotypic effect in cotton in general, so you do get bigger cells in the polyploids. But that experiment hasn't been done, nor the analogous ones for, say, nitrogen and phosphorus use, demands, and efficiency, which are the other things when you have larger genomes, not just cytochrome oxidase, so it's a whole nother dimension to that.
Yeah, kind of like piggybacking off of that idea, like, it seems like the nitrogen and phosphorus demands for making so much extra, like, genetic content would be a lot. Have you seen anything about, like, connecting soils that are maybe rich in those elements, or maybe increased level of, like, mycorrhizal associations in order to be getting those elements?
I'll answer the second part first. We know nothing about the mycorrhizal associations. Shockingly little about the wild plant autecology in general. And I mean, I guess I would say we don't know anything. We don't know how to answer your question. Even we don't. The whole thing about so the idea is that bigger genomes place high demands on phosphorus and nitrogen, especially phosphorus. Plants hold on. My friend Andrew Leach says plants are just going to grab onto every pea they can get and hold onto it tight. Phosphorus is pretty rare out there, and so nucleic acids place a phosphorus demand on the plant. So the idea is that larger genomes require more phosphorus. But there's a lot more RNA in plants than DNA. And so you can't assume that a cell is a cell is a cell and is making has the same amount of RNA and same amount of RNA turnover. And so that becomes a complicating factor. Those experiments are possible in good systems. Brand new paper came out yesterday or the day before? Yesterday, I think, in the New Phytologist on grass from Andrew Neely Leach on genome sizes and grasslands. I don't know, maybe you saw that, yeah.
I'm curious about novelty that comes out in the course of polyploidization, and something that being a zoologist I might not think about in the same way. And also just, I guess, piggybacking on maybe the Nobel Prize in protein folding chemistry that just came out in predicting these things. Do these kinds of events actually give rise to fundamentally new protein shapes or structures, or other, like the opportunity to mix genomes together? Is it giving plants an opportunity to access some kind of novelty that isn't present through other kinds of processes?
I'm thinking you've got me thinking about your very good question. I'm trying to think, are there any, do we know of any protein complex in any allopolyploid plant, because autopolyploidy is a little different, where a novel protein complex has arisen that didn't exist in either progenitor or diploid? Can any of you help me out?
I don't think I've ever heard
Lovely question.
of it. Yeah.
But polyploidy is so common in animals, you should go figure it out there. Yeah. In fact, it's super common in us. Cotton makes my octoploid cardiomyocytes beat a little faster, right? Yeah. By the end, it's a huge topic in cancer biology. Over half of all cancers have polyploidy, and a big chunk of the others have aneuploidy in them as well. Gene balance turns out to be such a big deal, and that's how gene fractionation is utterly mystifying to me. When gene balance is such a huge deal, you have to cross the Rubicon. How are you going to do it? I mean, you'd seem there's huge fitness valleys everywhere that have to be surmounted. My answer to all this is, of course they're surmounted. NE is small. NE is 1 in many cases. If NE, you can do anything. With NE equals 1, you can do anything. As long as you don't get extinct, right? Then you're the only game in town, and so now you've got a slightly more fractionated genome. Just forget about selection. Then it's easy.
So I'm intrigued by the multiple independent events of allopolyploidization. Do you see any, or have you detected in the cotton system parallel patterns of bias in retention of genes? Any kind of parallel sort of fractionation going on across the independent events?
The numbers are small enough overall, when you lose about 1,200 genes across both genomes, and you've got short tips with very little gene loss, the opportunity to look for parallelisms in gene loss are limited. That said, we have not looked with that question in mind. Thank you again. I should be taking notes. Oh, I'm so glad this is being recorded. Who else wants to? Go ahead, John, then Ingrid. Wait, John, even with my hearing aids, I can't hear you.
Tell us about how chromosome number has not kept up with the number of polyploidy events throughout the history of the flowering plants. But I'm thinking about ferns, where you have very high chromosome numbers in ferns. And if that's related to polyploidy, and why ferns don't lose all their chromosomes.
So I think you're right, and I think that's the conclusion. In fact, I think there was a recent paper that reached that same conclusion, that ferns have had the polyploidy, they just don't condense their chromosomes so much following polyploid events. Is that, you saw that too? So, I get that right. I don't know, we don't know why. There's hypotheses regarding why chromosome number should fall in general, but why that should differentially affect ferns and angiosperms? You have an idea?
There is a hypothesis that because of additional ways of inbreeding in ferns, that polyploidization is likely to be more frequent than in angiosperms. And so the generation of the higher numbers may simply be higher as opposed to the other process we just mentioned, namely the
lack of condensation.
Like, to what extent are they sexual? It might have to do with the relative importance of recombination events. Because if recombination is important versus not, the number of chromosomes will affect the size and number of chromosomes will affect the total number of recombinations given the size of a genome.
Yeah, I don't know if the DNA breakage repair machinery in ferns is any different than in angiosperms, which would allow for the kind of recombination events that would allow for that would result in chromosome condensation. I don't know if those two dynamics are different.
Look at their protein structure.
Yeah. Pardon me? I mean, even in plants that have like diffuse centromeres, chromosome numbers are all over the map. Yeah. I don't know if they're different either. Yeah.
Any other questions?
Did you have a question, Ingrid?
Oh, I was going to ask what was going on with that chromosome number 6 of the cross of the rice that you had showed. It was like it was all one genome, and the rest of them were really a mixture of things.
No idea.
Like, what's on chromosome 6?
With the experiments now in generation 12 or 13,
Oh.
and it's amazing that we're getting apparent selection for certain combinations, and a lot of phenotypes that would clearly be visible to selection in a natural setting, like flowering time, grain size, that sort of thing. Yeah, I think homologous recombination is really cool and going to be important. Yeah.
Well, if there are no other questions,
Should I cut it off? Should I cut it off?
We'll cut it off, but people can continue to
Okay.
discuss.
Come on up if you want to. Thank you. Thanks.